Good morning, everyone, and thank you for having me here today. I have nothing to disclose. Our studies of former preterm lambs include analyses of the heart. Our rationale for including the heart is that important comorbidities of evolving bronchopulmonary dysplasia in chronically ventilated preterm human infants are pulmonary hypertension and increased muscularization in pulmonary arterioles revealed by autopsy studies. If pulmonary hypertension persists, it may lead to lifelong consequences related to right-sided heart failure. With this knowledge, we asked the question, do former preterm lambs, abbreviated FPT, have these same characteristics? For context, using our chronically ventilated preterm lamb model, we have shown that the same pathophysiologic changes happen in the lung during the period of ventilation. This graph shows that pulmonary vascular resistance decreased from week of life 1 to week of life 3 in term reference lambs that were not ventilated, as should be the case for normal term neonates. By comparison, the black bars in the graph show that pulmonary vascular resistance did not decrease in preterm lambs that were supported by invasive mechanical ventilation over the same postnatal time period. The difference at 3 weeks for the preterm lambs is statistically significant compared to the matched term reference group that was not ventilated. The bottom graph shows for the same groups of lambs that muscularization of pulmonary arterioles persisted in chronically ventilated preterm lambs, indicated by the asterisks, compared to the matched reference group at 3 weeks. Lamb hearts were examined at 2 and 5 months postnatal age. Weaning from milk occurs at 2 to 3 months postnatal age in sheep, which is equivalent to 1 to 2 years in humans. We interpolated 5 months postnatal age in sheep as equivalent to 6 years postnatal age in humans, using the developmental landmark of puberty onset in sheep at 8 months of age. This graph shows that persistent muscularization of pulmonary arterioles occurs in the lungs of former preterm lambs. Results are shown as the mean, plus or minus 1 standard deviation for a sample size of 5 per group. Mean smooth muscle area in micrometers squared is significantly larger in former preterm lambs at 2 months corrected postnatal age compared to the age-matched term control lambs. Muscularization persisted at 5 months corrected postnatal age among former preterm lambs compared to their age-matched term control lambs. This slide shows that right ventricular free wall thickness postmortem is significantly greater in the former preterm group at 2 months corrected postnatal age compared to the age-matched term control lambs. By comparison, left ventricular free wall thickness was the same between the two groups. These results suggest that only the right ventricle is affected in former preterm lambs, at least at 2 months corrected postnatal age. Results for former preterm lambs at 5 months were not shown because these hearts are being imaged by diffusion tensor imaging, including measurements of volume of the right and left ventricles, which will provide volumetric estimates of Fulton's index. Our lab uses dynamic in vivo diffusion tensor imaging of normal term control lambs combined with post-mortem confocal imaging to track cardiomyocyte development in lamb hearts. So our results show and give an estimate for location, orientation, and anisotropy of cardiomyocyte fibers in DTI datasets and confocal histology. We combine both of these imaging modalities to track and quantify cardiac architecture during normal development. We are also hoping to use these imaging modalities to characterize progression of cardiac remodeling in former preterm lambs as they age. This presentation today will provide some of the confocal results. So our objective is to identify the long-term impact of invasive mechanical ventilation on cardiac architecture and histomorphology in former preterm lambs. Fetal lambs were exposed to antenatal steroids before operative delivery at about 128 days gestation, term being about 150 days in sheep. Preterm lambs were intubated and treated with surfactant and caffeine citrate. They were supported by invasive mechanical ventilation for 3 days, extubated to non-invasive support for 3 days to transition them to be weaned from all respiratory support. The former preterm lambs remained housed in our laboratory where they were pair-fed as they grew for 2 or 5 months corrected postnatal age. The lamb hearts were fixed at ventricular diastole and cut in cross-section. We used histochemistry for confocal imaging, utilizing wheat germ agglutinin to stain the extracellular matrix, Griffonia isolectin B4 to visualize capillary and endothelial cells, and TOPRO3 to visualize the nuclei. And then we used ImageJ and MATLAB to analyze these staining. This image shows fluorescent staining in the right ventricle from a 2-month term control lamb on the left and a former preterm lamb on the right. Again, green is wheat germ agglutinin, which is outlining the contour of each cardiomyocyte by staining the extracellular matrix, and blue is TOPRO3, which is staining the nuclei. All images are shown at the same magnification. The bottom images show the staining for the left ventricle of the same lambs. Cardiomyocytes in the right ventricle of the former preterm lamb do appear narrower than those in the term lamb. This slide is organized in the same way as the previous slide, except these images are taken from 5-month preterm lambs. Cardiomyocytes appear similar in both groups of lambs. This slide graphically shows cardiomyocyte length in micrometers as a surrogate for stereologic quantification of cardiomyocyte hypertrophy. The results are shown as mean and standard deviation for sample size of 4 to 5 lambs per group. At 2 months corrected postnatal age, former preterm lambs had significantly shorter cardiomyocytes in both the right and left ventricles compared to the respective age-matched term control lambs. This difference did not persist at 5 months corrected postnatal age for either ventricle. This slide graphically shows that cardiomyocyte width was not different between either group of former preterm lambs and their respective age-matched term control lambs. One of our colleagues at Monash University in Melbourne, Australia, is using stereologic techniques to quantify the volume of cardiomyocytes as the gold standard indicator of cardiomyocyte hypertrophy in these same hearts. We are also quantifying hyperplasia. However, these results are not available at this time to report. The next portion of our analysis involves quantification of capillaries in the myocardium. This image again shows wheat germ agglutinin in green and Griffonia isolectin in red, co-localized in cross-section of the left ventricle of a term control lamb at 2 months postnatal age. Because the two colors yielded yellow, visualizing the capillaries is difficult, therefore we'll show only capillaries with the red immunofluorescence. These representative images are from hearts of lambs at 2 months postnatal age. Capillary profiles are shown in cross-section for the right ventricle of a term control lamb on the left and a former preterm lamb on the right. Likewise for the left ventricle. These are from hearts of lambs at 5 months postnatal age. We are currently in the process of writing a code for MATLAB so that we can quantify the density of capillaries per unit area of myocardial tissue in these lectin-stained tissue sections among the groups of term control and former preterm lambs. So in summary, cardiomyocytes are shorter in the right and left ventricle of former preterm lambs at 2 months corrected postnatal age compared to term control lambs. However, shorter cardiomyocytes did not persist in the right or left ventricle at 5 months corrected postnatal age. So we conclude that hearts of former preterm lambs have some architectural changes that may be manifestations of evolving pulmonary hypertension at 2 months corrected postnatal age. However, the changes do not appear to persist at 5 months corrected postnatal age. So we have several steps that we still need to complete, including increasing the sample size per group, as well as relating these results to diffusion tensor imaging results and direct measures of pulmonary artery pressure and calculation of cardiac output and pulmonary vascular resistance. Lastly, we want to increase the number of days of invasive mechanical ventilation to determine whether cardiac structural changes persist longer in life. Thank you. Any questions?
One of the other major findings of your temporize many adverse effects associated with invasive ventilation. Have you guys had the opportunity to look at that in this model?
We have not with the confocal and diffusion tensor imaging analysis. We've started doing diffusion tensor imaging analysis on high nasal HFNV lambs, but those results aren't published yet and they're not readily available at this time.
Thank you.
Yeah.
All right.
Thank you.
So much, Cara. Okay, Cara's. All right. So our next presentation is from Sydney Bowen. She graduated from the University of Utah as an undergrad, and she's been accepted into graduate school at the University of Colorado, so will be leaving us to go and do an MS in epidemiology. So her talk today is on the structural and developmental changes in the brain of normal and former preterm lambs.
Thank you and good morning. I have nothing to disclose. So MRI is useful for human neurodevelopmental studies, and it contributes to the discovery of mechanisms that may be therapeutically targeted. These discoveries highlight the need for realistic animal models of brain structure and development in preterm neonates where additional mechanisms and therapies may be evaluated. Though few studies have looked at the structure and development of large animal brains in the setting of premature birth, including following invasive mechanical ventilation. For this study, 2 groups of lambs had magnetic resonance imaging done according to this timeline. The first group is normal lambs born at term and managed by MRI at about 1 week of life, then at 1 month, and at 2 months and 5 months. So, as we saw in Kara's presentation, these are the same lambs that we're talking about. So, 2 and 5 months postnatal life in sheep is equivalent to 1 to 2 years, and then 6 years in humans, respectively. The other group is our former preterm lambs. These former preterm lambs were made by delivering fetal lambs prematurely at 128 days gestation. They were managed by IMV, then transitioned to non-invasive support, and then weaned, like Kara said previously. Term and former preterm lambs were pair-fed used milk for 2 to 3 months until they are weaned to grass and grain. Lambs are transported to our institution's Human Imaging Center for in vivo imaging. Structural scans were T1 and T2 weighted at the resolution shown. Raw T1 and T2 MRI images were post-processed, corrected for rotation and orientation, then normalized and stripped of non-brain tissues. Image processing was done using software programs in MATLAB. This picture up here shows a scan of the brain of a 3-month-old lamb that was a template image to which all other brains were co-registered. This segmentation process gave images such as these for each scan that showed, for each voxel, the percentage of gray matter, white matter, and cerebrospinal fluid in the voxel. For this presentation, gray matter segmented image was used for remaining statistical tests. We used newly published high-resolution MRI mapping data by our collaborators, Drs. Ella and Keller, who are at the University de Tours in Noisy, France. They created neuroanatomical regional parcellation maps for normal sheep, which we used to map the brain of normal developing lambs and preterm lambs that were ventilated postnatally. This slide summarizes the postnatal age and days of the 52 lambs for which we obtained T1 and T2 images. The age range is from 1 week old to 6 months old, which is 180 days. 66 scans were made for 42 normal term control lambs of varying postnatal ages, shown up by the blue dots. 15 scans were obtained for 10 former preterm lambs that were mechanically ventilated in the first days of preterm postnatal life. These are shown by the red dots. Repeated imaging was done for some lambs, shown by the colored lines connecting the dots. This slide will graphically show mean gray matter density for 50 cortical gray matter regions in the brain of lambs using both hemispheres. We used a linear mixed effects model that incorporates postnatal age, preterm status, and individual lamb for random effect. In this graph, blue dots and lines are for normal term control lambs, whereas the red dots and lines are for former preterm lambs. The lines indicate repeated MRI scans for the particular lambs. Shaded areas are 95% confidence intervals for group means. This composite slide graphically displays mean gray matter density during the two developmental epochs, birth to 2 months postnatal age, and after 2 to 5 months postnatal age. Modeling was done from T1 weighted images for the left and right hemispheres shown on the left, and T2 weighted images shown on the right. The slice plane is through the parahippocampal region of the brain. The thick yellow line in each graph shows the slope of results for the first 2 months of postnatal age. Mean gray matter density increased during this period. After 2 months postnatal age, mean gray matter density plateaued, shown by the green lines. This composite slide graphically displays mean gray matter density for the cingulate region of the cortex, from birth to 2 months postnatal age on the left, and from 2 to 5 months postnatal age on the right. The main point is that gray matter density increases during the first 2 months of postnatal age, shown by the yellow lines again, and then plateaus from 2 to 5 months, which is again shown by the green lines on the right. We illustrated in the previous and this slide that the hippocampal and cingulate regions of cortical gray matter, because they are important for memory and learning and attention, including to novel objects, which sets us up for Caitlin to present May's analysis of the lambs that we did the brain scans on in the next presentation. These serial, color-coded, T1 and T2 weighted slice images show statistical correlations between age and T-statistic for gray matter density for all lambs, including our former preterm lambs, from birth to 2 months postnatal age. The color key in the bottom right of both panels shows the T-statistical levels for gray matter density in the cortex. These regions show that the greatest increases in gray matter density are yellow through red. These white ovals identify the parahippocampal region and the temporal lobes and cingulate cortex near the midline in particular. These T1 and T2 weighted slice images are for the same 52 lambs, but from 2 months to 5 months postnatal age. This shows that none of the regions have significant changes in gray matter density, indicated by the blue and pale orange colors, which show subthreshold T-statistic for change with age in each region. These T1 weighted slice images reveal the impact of former preterm lambs on gray matter density. That is, the colors are for the subset of former preterm lambs for the two developmental epochs, birth to 2 months on the left and 2 to 5 months on the right. In both panels, blue and pale orange colors suggest that gray matter density may be lower among former preterm lambs from 2 to 5 months postnatal age. These white ovals identify parahippocampal and cingulate regions that have apparently lower gray matter density due to the former preterm lambs. While these findings are not yet statistically significant, the impression of decreased gray matter density may be emerging as a long-term consequence of preterm birth and mechanical ventilation. We conclude that gray matter maturation is active from birth to 2 months postnatal life in sheep. The biggest changes are seen in the parahippocampal and cingulate regions from birth to 2 months. Differences in gray matter density related to preterm birth and ventilation are suggestive that cortical gray matter development may be less in former preterm lambs. The latter initial results require many more former preterm lambs for statistical assessment in the contribution of premature birth and ventilation to brain development. Thank you.
Any questions?
So, can you, in the lab, do you have fun tests that you can put the lambs through after you do this imaging to see if they learn more or less than term lambs?
Yes.
Or is she getting to that?
Yeah, Caitlin is just about to present that. No, perfect.
Other questions?
Oh.
So you posed a really important question in the last question, which is it being preterm or is it mechanical ventilation or are they synergistic? How do you propose to dissect that? Or how would you want to go about dissecting it?
Great. So we are currently working on essentially testing the dose of invasive mechanical ventilation. So we have preterm lambs that we developed or that we've delivered at the same early gestation, but some are on IMV for 3 hours, some are for 3 days, and some are for 6 days. So we're hoping that then we can see if it is IMV or if it's just being preterm that's showing these effects in the brain. Thanks.
Thank you. So our next presenter is Caitlin Jarret. She was an undergrad student from the University of Utah who recently graduated and is planning to apply for PA school next year. She will be presenting on former preterm lambs that have neurodevelopment impairments later in life, so we'll have the whole physiological story of the lambs.
Thank you. Good morning. Okay, I have nothing to disclose. Long-term neurodevelopment is a concern for survivors of premature birth and invasive mechanical ventilation. About 60,000 infants per year are born in the U.S. with very low birth weight, that is, birth weight of 1,500 grams or less. 25 to 50% of these preterm neonates will have cognitive, behavioral, attentional, or socialization deficits later in life. Brain injury in these neonates may be cystic or non-cystic periventricular leukomalacia that may be focal or diffuse. Brain injury also may be intraventricular hemorrhage. However, the pathogenesis of brain injury to chronically ventilated preterm neonates is incompletely understood. Our laboratory is identifying pathogenic mechanisms leading to brain injury in preterm lambs that are supported by invasive mechanical ventilation, abbreviated IMV. Our studies show that IMV leads to non-cystic, non-hemorrhagic diffuse brain damage, including cortical gray matter regions such as the hippocampus. Also, loss of cortical white matter occurs as shown on the next slide. The point of this graph is to show that 21 days of IMV lead to significantly less cortical white matter area normalized for brain tissue area. This is a former preterm lambs. Sorry, I'm trying to get which is the red bar. The other preterm lambs that were managed by 21 days of non-invasive support, abbreviated NIS, is the black bar, and unventilated term lambs is the gray bar. Manifestations of loss of white matter are illustrated in this slide, which shows a postmortem diffusion tensor image of the brain of a normal, unventilated fetal lamb. The rainbow of colors reveal ascending, descending, and crossing fiber tracts of a 1 millimeter thick slice. This picture shows a similar view of the brain of a preterm lamb that was supported for 3 days of IMV. The two yellow ovals draw your attention to crossing tracts that are less obvious in the cerebellum and the anterior horn of the corpus callosum. Although not shown in this slide, quantitative histological analyses of these brains reveal that IMV is associated with more apoptosis and less proliferation of neurons and glial subtypes. Based on the background that was just presented, our aim was to identify the impact of IMV on neurodevelopment of former preterm lambs compared to term control lambs. Our hypothesis is that former preterm lambs will have neurodevelopment impairments that persist later in life. The fetal lambs were exposed to antenatal steroids before operative delivery at about 128 days gestation, term being about 150 days. The preterm lambs were intubated and treated with surfactant and caffeine citrate. The preterm lambs were supported by invasive mechanical ventilation for 3 days. They were then extubated to non-invasive support for about 3 days to transition them to supported spontaneous breathing before being weaned from all respiratory support. The former preterm lambs remained housed in our laboratory, where they were pair-fed as they grew. The lambs were tested at 2 and 5 months postnatal age. We selected these postnatal ages because weaning from milk occurs at about 2 months postnatal age for lambs and 1 to 2 years postnatal age for humans. We interpolated 5 months postnatal age in sheep as equivalent to 6 years postnatal age in humans, using the developmental landmark of puberty onset in sheep at 8 months of age. We chose both postnatal ages because they are used clinically to predict lifelong neurodevelopmental outcomes in former preterm lambs. The demographic characteristics of the lamb groups are summarized on this slide. Former preterm, abbreviated FPT lambs, are 6 in number, with equal number of females and males. Control term, abbreviated T, lambs were 14 in number, with 9 females and 5 males. Term lambs were not ventilated and served as normal controls. All lambs were pair-fed used colostrum followed by used milk. After weaning from milk, the lambs continued to be fed by bottle and given grass and grain. At 2 months corrected postnatal age, both female and male former preterm lambs weighed less than respectively matched control term lambs. Postnatal growth restriction persisted at 5 months corrected postnatal age for the preterm lambs. Because we receive lots of sheep jokes, we hasten to point out that the adult sheep are used for neurodevelopment testing using tests similar to those of rodents. We adapted tests for novel objects, reflective versus non-reflective surfaces, and maze. Neurodevelopmental tests are at 2 months and 5 months postnatal age, which is the corrected postnatal age for former preterm lambs. After habituation to the test room, tests are on sequential days. Tests are repeated as trials of 10 minutes each, separated by a 30-minute rest period. Lambs are then recorded while in the testing room. For the novel object and mirror test, we measure the time to the first encounter, the number of encounters, and the total time spent at an object. For the maze test, we measure the time it takes the lamb to locate the reward. The next 2 slides will show video clips of selected results for tests for which statistically significant results are detected at this point of our study. The following 2 videos will show the maze test. The videos are of lambs at 5 months postnatal age, which is important to identify because this means that the lambs did the same test 3 months earlier when they were 2 months postnatal age. Each video clip begins with a title that identifies the lamb group and test. The first pair of videos will be for the mirror test, in which reflective and non-reflective metal plates are used. Let's see if we can get this started. Okay, so this is a term lamb at 5 months doing the mirror test. So the lamb is in the room. The lamb spends much of its time at the reflective surface, in which you will see the reflection of the lamb's face. So this is a former preterm lamb at 5 months doing the same mirror test. This video clip starts when the former preterm lamb is in front of the reflective surface. If you watch closely, you'll see that the lamb right now headbutts the mirror. After the headbutt, the former preterm lamb moves to the non-reflective surface where it remained. The next pair of videos will be for the maze test, in which the lamb navigates through a maze in order to find a reward. So this is a term lamb at 5 months participating in the maze test. The lamb will enter the room from the top right corner. The video clip pretty much says it all for the control term lamb. The lamb reached the bottle of milk in about 5 seconds. So this is a former preterm lamb participating in the same maze test. However, this video clip starts when the lamb has already entered the room. The video clip shows a former preterm lamb that has a lot of difficulty navigating the maze to find the bottle of milk. Almost 100 seconds transpired before the lamb found the reward. This graph will present the results on the y-axis for time in seconds to find the bottle of milk. The x-axis identifies the former preterm and term control groups of lambs at 2 and 5 months postnatal age. Scatter plot results will be shown with median and interquartile statistics superimposed on the data points. Each black dot represents a lamb in each group. The yellow line is the median for each group, and the black vertical lines are the interquartile limits. At both 2 and 5 months corrected postnatal age, former preterm lamb groups took similar period of time to find the bottle of milk as the respective matched term control lamb group. This graph also shows that 5 months corrected postnatal age, the former preterm lambs have a wide range in the maximum time to find the bottle of milk, which is why the statistical difference was not detected with the sample sizes at this time. This graph similarly presents the data in time and seconds spent at the non-reflective surface. At both 2 and 5 months corrected postnatal age, former preterm lamb groups spent significantly more time at the non-reflective surface, which is indicated by the asterisks, than the respective matched term control lamb group. This graph also shows that the former preterm lambs have a wide interquartile range for the maximum time spent at the non-reflective surface. In summary, normal term control lambs learn and remember. Former preterm lambs appear to have learning and memory deficits. We suggest that 3 days of invasive mechanical ventilation of preterm lambs is sufficient to cause neurodevelopmental impairment that persists later in life. We continue to add more lambs to both groups because of the large variability displayed by the former preterm lambs. Thank you so much. Are there any questions? Yes.
So what neurocognitive function is the mirror test supposed to reflect or represent?
So it's kind of fear. I think it's the socialization and fear aspect. So if they're seeing themselves in the reflective surface, we're just kind of seeing if they are scared of it. We've found that a lot of the control lambs seem to hang around the mirror more than the former preterm lambs. It's just a fear aspect. But other than that, I'm not too sure. That's a good question, though. Yes.
Sorry, I missed this. So how preterm are they, and how does that translate?
So
Equivalent to a human preterm.
so we deliver them 128 days gestation, which is about 28 weeks gestation in human.
You have a group that's surviving but not ventilating?
So we don't have a way to resuscitate them right now without ventilation.
So if they're not ventilated, they die.
Right now we haven't come up with a way to resuscitate them yet. Yes.
So we actually do have a group that we ventilated only for 3 hours in a way, because that's the user under general anesthetic, and that's the amount of time that it takes us to get the lamb awake and resuscitate it. But we are currently working on a protocol where the lambs are not intubated and not given surfactant, and we're trying to resuscitate just using a mask that we're going to compare to these same lambs.
Yeah. So we haven't done a test on those lambs yet,
Well,
but
28 weekers who need ventilation are going to get ventilation. So I guess the question is really what you're trying to do is there a better way to keep them alive?
yeah, and
Yeah, and that's what we're trying to look at,
Yeah.
the 3 hours or 3 days, the no resuscitation, and then also the longer IMV.
yeah. Yes.
So the maze test showed a lot of variability. I guess the maze test is measuring memory and learning, kind of like sending angry tweets early in the morning. But
Yeah, right now we just don't have a big enough sample size. We only have in our former preterm lamb group, we only have like, I remember saying it's like 9 lambs. So we're trying to, how many is it? 6. So yeah, we're trying to make the sample size bigger. We think that's part of the reason why there's so much variability right now. But once we add more lambs into that group, we think that there's going to be definitely less variability. So I think that's.
The other point that Caitlin would also like to make is variability for those 2 lambs that we saw in that group. They are the same outliers when we look at their lung function, at their pulmonary arterial pressure. So it's the same 2 lambs that are outliers. And what's interesting is that not all preterm babies have poor outcomes. There's some that do well and some that don't do well. And what we're finding is that the former preterm lamb model recapitulates the human disease process. And what we're interested in are what's different with those outliers.
Yeah.
Yeah, and that's sort of exactly what I was digging
Yeah.
at, because it might give us one insight.
And we're looking at, in our hypothesis is that there's some epigenetic modification that's going on. And so that's the route that we're looking at in the frozen and the fixed samples that we have of both of the brain, the heart, the lung, the kidney, and the liver.
How inbred are your lambs? Is there much genetic variability?
The lambs are bred at a very large sheep ranch, so there's not much inbreeding at all.
Yeah, there's one more. Are we done with questions or?
Yeah, no more questions. We'll move on if you can catch up with Caitlin after the talk. All right. We'll move to the last speaker this morning, and this is Matt Linakis, who is actually in my group and in his hopefully final year of his PhD. Hopefully will be depending later this year. Yes, the end is in sight. So Matt is going to talk to you this morning about acetaminophen glucuronidation in neonates, so sticking to our neonatal theme. So Matt.
Thank you, Dr. Sherman, and thank you to the organizers for having me here to speak today. And thank you all for coming. I'm actually pretty excited to be able to give this talk to you guys and to this crowd who are a little bit more clinically oriented than I'm typically used to giving presentations to. So I'd love to get feedback at the end if you have any. But so what I'd like to talk with you about today is a specific genetic polymorphism that we found to be associated with decreased acetaminophen glucuronidation in neonates. So to start, I have nothing to disclose. I had wanted to give a quick background as to why I'm here, although Dr. Sherwin kind of already did that. I am a PhD candidate in pharmaceutics and pharmaceutical chemistry, but the reason I am here speaking with you today is because Dr. Sherwin is my primary mentor, which means that I'm based in clinical pharmacology, Department of Pediatrics, and so a lot of my research does take a pediatric bent to it, which I like. So. Today, in terms of the professional knowledge gap that we'd like to address, we're looking at the effect of genetic mutations and metabolizing enzymes on the disposition and pharmacokinetics of drugs in general, but today acetaminophen in neonates because it's currently unknown. And so at the end of this presentation, we'd like to kind of be able to identify influential genetic variants of enzymes important in acetaminophen metabolism in neonates, and in particular, how the interplay between these genetic polymorphisms and the ontogeny. So when you add the development of some of these metabolizing enzymes in the neonatal population on top of these genetic variants, it makes for a very complicated picture. And so we're looking to start trying to deconvolute some of that. I don't need to tell this to this crowd, but pain pathways are intact and functional in neonates. It's always a little bit surprising to some people how frequently it was that this kind of came into the picture. And so because of that, we occasionally have issues where if there's inadequate pain management, that it can lead to short-term complications and additional long-term sequelae, including neuromorphological effects, altered pain thresholds, impairments of cognition, perception, or motor function. And so one of the things that has relatively recently come onto the scene then to deal with this is intravenous acetaminophen, which will be abbreviated APAP for the remainder of the talk. So acetaminophen, as you all know, is an analgesic and antipyretic. The specific intravenous IV formulation did get FDA approval only back in 2010 for adults and children 2 years and older. Obviously, there are a number of benefits to this formulation, so it can avoid or at least minimize adverse effects of opioids and other NSAIDs that may be used in these populations. And because it's an IV formulation as opposed to the oral formulation, it presents with a much more predictable bioavailability and onset of action. Unfortunately, what is unknown in this particular case is how it should be ideally dosed in neonates, particularly as it pertains to some of the pharmacogenetic information that we're going to discuss today. So just to give a quick background then on the actual pharmacogenetic information we're interested in, acetaminophen is primarily cleared by hepatic metabolism, and in that it's got three major pathways by which it's cleared. And I don't want to stick on this too long, but I do want to give a little bit of background as to those important metabolizing enzymes. So the first pathway is the glucuronidation pathway. And so this is this one up here. And the important metabolizing enzymes for glucuronidation are the UDP glucuronyl transferases, or UGTs. And so in particular, we have UGT 1A6 and 1A9 are going to be two of our most important metabolizing enzymes. We've also got a little bit of information about 1A1, but that one's not going to be as important per this project. The interesting thing about UGTs in this population is that it's well documented in the literature that UGTs do have a fair amount of development and ontogeny that's related to them. And so when we start talking about that interplay between ontogeny and genetics, that may come into play a little bit. The second pathway, the sulfotransferases, or SALTs, are thought to not have as much of a developmental effect. So typically, by the time a term neonate is born, and in fact, for the most part, preterm neonates, that pathway is more or less considered to be fully developed. Because of that, going into this project, it was kind of hypothesized that this would be the pathway that we saw the genetic variability would make the most difference, because we expected this to be the major pathway in neonates. Finally, not necessarily as important for genetic variability directly, although also very important for a reason I'll explain in a second, was the cytochrome P450s of the CYPs. In particular, we have information on CYP2E1, which is the major pathway, major enzyme for metabolizing down the oxidative pathway. And the reason that this is important is because the oxidative metabolites, and in particular MAPQI, is the toxic metabolite. And so what happens in the case of acetaminophen overdoses is that these other pathways tend to get saturated, particularly the sulfation pathway, and then it starts shunting towards this cytochrome P450 pathway and generates an increased amount of toxic metabolite. And because of that, this is why we want to be very careful in terms of if we do have any reduction in one of these pathways, we could potentially end up seeing a shunting of metabolism towards these other pathways. So then this study explored the effect of 17 of 21 total collected sequence variations in 5 metabolizing enzymes on acetaminophen glucuronidation, sulfation, and oxidation. And the reason that we do have a couple that were collected that we didn't manage to test is because we either saw no genetic variability or we didn't have enough information to actually be able to test those. So quickly on the study design, I'm not going to go through all of this, but I did want to put it up here. So for inclusion criteria, we are looking at infants less than 28 days old. So we're looking at neonates with an indwelling arterial line. We had a number of exclusion criteria related to comorbidities. That was simply for pharmacodynamic reasons. And then we additionally split these patients out into preterm with a gestational age of less than 37 weeks, although we also split them out further, less than 28 weeks being extreme preterm, and then term gestational neonates of 37 weeks or greater. So this is the demographic information for the 33 neonates we had in our study. We had a medium body weight of about 2.7 kilograms, medium postnatal age of 6 days. You can see we've actually got a pretty even split between our term and preterm neonates of 17 or 52% were term, and then 16 were preterm or extreme preterm. Another interesting thing, and the reason I had the primary indication for intravenous paracetamol, I'll get to that in a second, but we do have about half of our patients receive that for postoperative analgesia versus about half received it for medical conditions. And then we had a little over half of our patients were male as opposed to female. So what we did in this particular project is we actually built upon a population pharmacogenetic model that had been built by Dr. Sarah Cook for her PhD dissertation previously. And so we can see the general outline of this model right here, where essentially we have acetaminophen concentrations that we determine in the plasma. We have each of the three kind of main metabolite concentrations determined in the plasma, and then we have each of those additionally the amounts that were determined in the urine. And so Dr. Cook was able to generate a model based on that information. And so what we were able to do with the additional information we gained was we were able to then consider the impact of each of these metabolizing enzymes, so UGT1A1, 1A6, 1A9 on the glucuronidation pathway, SULT1A1 on the sulfation pathway, and CYP2E1 on the oxidative pathway. So what we do in order to test these things is we do a stepwise covariate modeling approach. So we essentially put each of these things in one step, consider using a log likelihood ratio test, whether it's statistically significant, and then if something is, we take the most significant thing and then move forward with it. If you're interested, I'm happy to talk to you a little bit more about that process afterwards, but given the limitations of time, what I just want to say right here is I know there's a lot on this slide, but I just draw your attention to this box right here. So what we did determine to ultimately be statistically significant at a high level, so a p less than 0.001, was this variation in UGT1A9, reference sequence 3832043. And essentially this is a T insertion mutation in the promoter region of the UGT1A9 gene. And so you can see we've got a pretty nice distribution of those individuals who are heterozygous for the wild type in this, those who are homozygous, and those who are heterozygous for the variant. This is the first important bit of information in terms of the pharmacokinetic model that we get excited about. It's statistically significant. I don't want to stay on this slide too long either. This is just a suggestion that the observed and the predicted concentrations in our model are lining up quite nicely. So in the idea of a perfect model, we'd see a one-to-one correlation. Obviously, you're typically never going to get that, and so, but we do see pretty nice along that line of identity in this case. This, I'd say, is the more pertinent information in this case, though. So we have our statistical significance, and that's great, but what does that mean in terms of the glucuronidation, that pathway that we have for metabolizing? So what we see here is if we have our homozygous variant, our homozygous wild type individuals, so sorry, the T9 up at the top, T9/T9, we have a glucuronide clearance of about 0.068. Then if we have any T10 alleles, so regardless of whether it's a heterozygote or homozygous variant, we see a reduction of about 41 or 42%. It is interesting that regardless of whether there is one or two variant alleles, it doesn't seem to matter. Those two individuals do seem to be about the same. But certainly the presence of at least one variant allele does seem to reduce the clearance via the glucuronidation pathway. Now, I do want to put a very quick disclaimer on this to say that just because there's this apparent decrease in clearance down this pathway does not mean that there should be any difference in dosing or anything like this. This is kind of one pathway, and we're still kind of trying to figure out what that means for the larger metabolic profile and the larger concentration of acetaminophen in general. But there's no indication right now that this suggests anything related to a change in dosing or anything like that. And so the information that we found in this study is consistent with at least one other study that suggests the presence of one T10 allele did alter the AUC of, in this case, a different drug, mycophenolic acid. And additionally, we need to go a little bit further into the SALT pathway. As I mentioned at the beginning, we expected that to be the more important pathway in this particular case. Right now, it doesn't look to be the case, but in delving into the data, we're seeing some interesting patterns like we see here, where we have for a copy number in SALT 1A1, there's a bit of a correlation with a high copy number, which you would expect to increase the sulfation clearance, but we actually see a decreased sulfation clearance compared to a low copy number, and I apologize, it's not coming out great. But it turns out that those with a high copy number tend to also be much lower gestational age, and so those types of things we're trying to tease out of the data right now. So finally, to conclude then, inclusion of UGT1A9 sequence variation information did significantly improve the fit of the previous PK model. And those findings indicate that the UGT1A9 genotype does impact the formation clearance of APAP to APAP glucuronide, but again, take that with a grain of salt. And then the reduction in that clearance is similar for heterozygotes as well as those homozygotes for the variant. This project includes a number of collaborators and a number of people who have worked on it, and I'd like to thank all of them for their support in this. And at this point, I can take any questions. Thank you.
Probably one question for Matt.
Yes.
That enzyme is also involved in bilirubin metabolism. By any chance note any abnormalities in bilirubin metabolism?
No, and unfortunately, we don't have that information available, but that would be a great thing to look into. Absolutely.
So what's known about the persistence of this variant? Acetaminophen is not, as you point out, it's rarely used,
Mm-hmm.
but it's used a lot for children.
Mm-hmm. Mm-hmm.
What do we know about that?
Well, and so in particular, this particular variant hasn't been kind of noted in those older populations, and my assumption would be that's largely related to the fact that by that point those pathways have matured. So it's a fairly rapid maturation of UGTs. I think it's something like 2 months. They're expected to more or less have most of their activity. And so typically by that point, you don't, that hasn't come out in the literature in the adults and older people.
Thank you,
Thank you.
Matt. And I think Dr. Bale has some awards.